So I'm going to tell you, as I say, as is shown in my title, something about molecules and microbes. I will not say very, very much about me, but you'll get it on the way probably. So we'll see what happens. So I want to start out by showing you this slide, which is a very famous slide, in which Selman Waxman, the discoverer of streptomycin, met Alexander Fleming, the discoverer of penicillin, in Waxman's lab, and Waxman is showing Fleming some tubes. Rumor has it that Waxman said to Fleming, "No, Al, they're not fungi." But this is a very famous picture because this really got the antibiotic industry started. And this is what I want to talk to you about, is a little bit first about the antibiotic industry, how it's grown, where it's gone, where it's going wrong, and some other things which were perhaps more recent work which will impact on this. So this is a historical slide in the sense that it's talking about history. It's not a very old slide, but it's showing you the origins and evolution of antibiotics and antibiotic resistance. In fact, this is a coevolution. These two classes of events, the discovery of resistance and the discovery of antibiotics and the discovery of resistance, really do go very closely together. And pointer here. You know, I have a problem here. It's interesting. I'm a mutant. I'm colorblind. So this doesn't work. There's nobody here who has a green laser by any chance, do they? All right. This is much better. Thank you. We have the discovery of penicillin at an early stage. As you probably know, penicillinase was discovered very soon afterwards. And then there are these various stages that take place during the antibiotic industry. Before antibiotics, as you well know, about the only thing you could do to prevent infectious disease was to wash your hands. And the interesting thing is that now, 60 or 70 years later, it's still the only way that you can stop yourself getting infectious disease. So don't forget Semmelweis. During the first 20 years of the antibiotic industry, it was an incredible period. Everything that was discovered turned to gold. Almost every compound that was isolated mostly from actinomycetes, but also, and this is forgotten a lot, that from bacillus in many cases, these compounds were very rapidly introduced and used in treatment. And then there are other stages during after the golden years when the antibiotic industry became sophisticated in various ways, in the sense that they began to consider pharmacology at one stage. They began to look at biochemical aspects of antibiotics, another stage, to look at targets. And then, of course, there was the disastrous period in 1990 when antibiotics were sought by genomic methods, by sequencing, and that was a complete and utter failure. Only one compound was found, and that was already known. And still now, we are in a period of disenchantment about the antibiotic industry and the whole role of the pharmaceutical industry in antibiotics. Now, there's one very key event in this whole process which tends to be forgotten, and that is in about 1965, the US formed the Food and Drug Administration Office of New Drugs. This was formed as a result of problems with certain drugs, and more particularly with pregnant women. And they formed this Office of New Drugs, which introduced regulations about toxicity. Before that time, antibiotics could be put on the market within a matter of months. And I think, I can't remember which one it was, there was one compound that was put onto the market about 4 months after it was first isolated. That is obviously no longer possible, and that was certainly stopped by the Office of New Drugs. And Frances Kelsey, who was the person who was responsible for that office, has a lot, well, we owe her a great thanks because of what she did for the drug discovery process. But it slowed it down, obviously. Another thing that is interesting is that in about the 1950s, the annual production of antibiotics was 50 kilos. In 2000, the annual production was 10 to the 6th kilos. So the production of compounds has gone up enormously, and we're in a situation where things have just gone on and on. And one of the problems is that resistance, which really began almost as soon as antibiotics were discovered, has increased all the time. And during this time was the lean years of antibiotic resistance development. The discovery of antibiotic resistance plasmids in 1965 complicated the issue because genetic transfer of antibiotic resistance was something that was never suspected. It was discovered in Japan, it was not believed in the United States, and it wasn't until somebody in the United States actually found a resistance plasmid that it was actually believed. So we have this complex and internationally intermingled pharmaceutical companies, drug companies, to the present stage where we have no new drugs. In the development of resistance, and this is clearly something that is very important, and it's clearly a very important task to try to control antibiotic resistance. But you can see from this slide that we're doing a very, very poor job of doing this. This is a graph of the evolution of the beta-lactamase enzymes, which give resistance to penicillins and related drugs, cephalosporins. And the MRSA was discovered down here in 1967. The first beta-lactamase was discovered shortly after that, and now we're up to about 1,000 different beta-lactamases. Different enzymes. Every one of these enzymes has evolved from the use of another compound or another set of compounds. So it's been a constant effect of evolution to a toxic drug which has led to the beta-lactamases, and we don't win. And MDM1, which I'm sure you know about, is right at the top. But there's already MDM2 and 3 and I believe 4. So it'll keep on going until we find another way of doing things. The oh, okay. I'm not very good with these tools. This reaction, the hydrolysis of a beta-lactamase, of a beta-lactam ring here, has turned out to be the most expensive enzyme or enzymatic reaction of all time. It's been something which the pharmaceutical companies have been working on, and they've done some wonderful things. They were able to modify the substituent on this ring so that it could become partially, the hydrolysis could be prevented, but the resistant organisms still come along. So it's not something that can be taken off very easily, and I think this is a most unfortunate thing. Now, if we go to the discovery of antibiotics, this is taken from David Hopwood's book, and David probably showed you this last year, and I apologize for showing it again, but one of the things I want to add, which I don't think David mentioned, is that the last antibiotic as such was spinosyn that was discovered. The last naturally occurring compound which was an antibiotic was spinosyn. Spinosyn is not for human use. It's used in animals. So that the human use antibiotics go back a little bit further. So we're doing very, very poorly in trying to come up with compounds that can actually take care of resistant organisms. So this is a question that needs to be addressed. if we are going to keep using antibiotics, we are going to be faced with resistance. And so we have to come up with some new ideas. And this is, these are some of the things that have been talked about. And the background, of course, is if we fail. This background could also be the French after the Wales beat them last weekend or something. But on the other hand, it demonstrates what happens. This is the Black Plague. It demonstrates what can happen. And one of the things that everybody is pushing for is new antibiotics. A lot of other things could be done, but it's proving to be politically very difficult to control antibiotic use, to ban non-human use. And so we're faced with the only option at the moment is try to come up with new antibiotics or modifications of old antibiotics. And this is very difficult. The companies just don't find things any longer. And obviously, as you probably know, the companies are not terribly interested in looking for something that they're failing at and is not going to make them very much money either. So we're in a difficult situation. And the IDSA, the Infectious Diseases Society of America, said that for the infectious diseases personnel, they want new antibiotics and they want them quickly. And in fact, they have a scheme, a plan, a grand plan, that 10 new antibiotics will be found by 2020. Now, when you think about that, it's ridiculous, because they proposed this a few years ago. It takes anywhere from 6 to 8 years to go through clinical trials. We're now in 2002. There are no new antibiotics on the scene. So we're not going to get 10 new antibiotics by 2020. We might get 1 or 2. And if we do, I think that will be a great thing, but we'll see. So I wanted to tell you now what is really the problem about the antibiotic business. Antibiotics have been worked on, as I've mentioned, for 50, 60, 70 years. But the problem is nobody has ever asked the question, what are these compounds doing in nature? It's been assumed from the very, very beginning that the compounds that were isolated from soil were produced by microbes to create a defense system against other microbes. This has never been proven in the environment. The environmental roles of antibiotics is very poorly studied and very poorly known. So the question is, what are these compounds doing out in nature? And this is really the only research that has been done on antibiotics over the past 50 years, which is it's fermentation research. So you take an organism from the soil with a doubling time of months that exists under low aeration, variable nutrients and carbohydrate, variable water, a temperature from 0 to 40, and a pH of 10, so 2 to 10, and the yields of compounds are really very low in the soil. They're so low that we can't detect them. So this is the basic thing, is what's happening in soil. And the most important thing is the doubling time of months. Now, when pharmaceutical companies isolate a strain producing an interesting looking compound, then they submit it to fermentation. And under the production in large-scale production, the doubling time is hours, nutrients are high, carbohydrate high, water is unlimited, etc., etc. The yield is in grams. You don't find grams of antibiotics in soil. And in fact, to my knowledge, nobody has ever isolated an antibiotic from soil and shown what its structure is. If anybody knows of such a thing, I'd like to know. And the other interesting thing is that the nutrients used by the pharmaceutical industry are amazing. One of the things that have always surprised me is that the kinds of things that are used to try to provoke an organism to make a compound which it doesn't normally make very much. And hydrolyzed rabbit's fur as a source of nitrogen is really creative. But it worked for some antibiotics, and but you can't deny that. The point is, it's hardly a scientific procedure, especially when you mix it with a bit of pork liver and asparagus juice. So antibiotic discovery, and antibiotic science was something which was not well discovered. So we have to think about what we can do, how we can learn more about antibiotics in the environment and what they do. And I want to introduce you to my favorite operon, which is the Lux operon, which comes from luminescent bacteria. The Lux operon has been developed in such a way that you can detect transcription of the operon by the production of light. So this particular operon is an ideal system for scanning for transcription activities of small molecules or anything you like. And as you will see, we have tested anything you like. So transcriptional activation can be looked at using this assay, and one can use transcriptional activation as a way to find small molecules. This is a good example of the type of thing you can see with transcriptional activation. Here's an e-test of erythromycin, which is placed on a Petri plate. You can see that looking at it under the camera, and this is the particular Lux construction. And you see that you don't see very much color. You look at it properly under the camera and you see this. Now this is a striking example, but it's not unusual, of what you can get when you put a bioactive compound on a strain which has an inducible Lux expression system. The important thing here is that this is the zone of inhibition right here. And you see that as you move out from the zone of inhibition, you get decreasing amounts of light. And these decreasing amount of lights indicate that even a long way from the highest concentration, you still get activity. So we're looking here at definitely minimal inhibitory concentrations. In fact, very minimal inhibitory concentrations. So the question then is, if this is the way antibiotics work in the lab, as detected by transcription systems, you have to ask the question is, how can we possibly find things in nature, or how could the pharmaceutical companies possibly have found things by using the kinds of gross methods that they used? The question now is, can we use this? Can we apply Lux expression for detecting new molecules? And together with Mike Surrett a few years ago, we made a random library of promoter Lux reporters in Salmonella. And we started off with about 7,000 clones. We could then screen these 7,000 clones. I don't want to go through what is actually done here, but we have them all in plates, and we can screen anything we like for reporter responses. That is not the only thing that's important. The most important thing is to say, if you screen for reporter activities, the question is, what reporters? Is it always one reporter, or are they specific reporters? And happily, they're very specific reporters. And again, this is a few years old, we were able to show that with most of the antibiotics that are used, one gets a bell-shaped curve. It's funny bell, isn't it? But it's sort of bell-shaped curve in which the environmental, what we believe to be the environmental aspects of the activity, these occur at very low concentrations. This is 0.1 of the minimal inhibitory concentration. And then when you get to an inhibitory concentration, you get a very rapid inhibition of transcription, except for transcripts enhanced by stress. So this is really the stress response here. So we would argue, and we've been using this as our basic principle, that the low concentrations of these compounds, it's receptor-mediated transcription, high concentration is target-mediated inhibition. So we've been using this and having a lot of interesting results from it. Most of these results we can't really explain at the present time, which is a good position to be in. I have, I can constantly get students who want to have a crack at this. So this is a very good example of the macrolide antibiotics. I'm sure you all know them. You know erythromycin, azithromycin, the other related compounds of pristinomycin and lincomycin. Now, these all act at the same place, virtually at the same place on the ribosome, but not identical. And this is one of the interesting things about this, because here's erythromycin. Azithromycin is a relative of this. And here we've plotted the responses of the Lux reporter assays for all of these antibiotics here against erythromycin. So anything that is similar to erythromycin falls on this line. And the only thing that really falls on this line is azithromycin, which is closely related to erythromycin. What is interesting are things like pristinomycin, which is sold as Synacid. It's used as a treatment for gram-positive infections. And Synacid is spread all over the place, but it's certainly not overlapping with what the effect is on erythromycin. And in this particular case, we've got tylosin. Tylosin is closely related to erythromycin, but it is not showing quite the same effect. It's more or less on the same line. Lincomycin, you will see, is falling in a different area of the box as well. So one can use this to say, well, we've got all these compounds that are acting at virtually the same place on the ribosome. They are inhibiting protein synthesis by essentially the same overall mechanism, but clearly by different types of mechanisms. And this is shown here, the kinds of things one can do. Here you have the 50S subunit of the ribosome. One has this group of compounds, macrolides, streptogramins, and lincosamides, which all act at the peptide synthetase center, but they act differently. We've looked at other compounds and find that they also can be easily distinguished. So if you can use the reporters, and you know that the compound have a target like the ribosome, then you can begin to ask questions about finding compounds which have different modes of action. And here's the 30S subunit, where, again, there's a clustering of the target sites, but these target sites are not identical. The target sites can be distinguished by using Lux reporters. There are some interesting paradoxes. This is one of the major paradoxes for us, and we're still studying it, and that's RNA polymerase inhibitors. Rifampicin and microsins, several microsins, are very specific inhibitors of RNA polymerase. They work, as far as one can tell, only on RNA polymerase, and they do a very good job, and they're successful drugs. The interesting thing is that rifampicin, at high concentrations, it stops transcription. It really does block transcription. Rifampicin at subinhibitory concentrations will do something different. It will cause signaling. It will affect transcription. And so we're seeing these two different effects. So it's the dose response curve that I showed you before, the bell-shaped curve. This is one side of the bell-shaped curve. This is the other one. And this is a paradox for us, especially because with rifampicin, it appears as if the region of the target in terms of activating transcription appears to be just upstream of the genes that are being activated in the Lux operon. We don't really know very much about it so far. The point is that we can use these assays to find compounds, and we can find compounds at very, very low concentrations, and that's what we need. So what's this telling us? For my money, it's telling us that we're looking for antibiotics in the wrong way. It tells us that we could do a much better job for finding these compounds, and also that we could do some better job of trying to overcome resistance by learning something about resistance earlier on. And here I want to move to a different area, and that is asking the question, what are subinhibitory concentrations of antibiotics doing in nature? Since they are subinhibitory levels in nature, they're doing something else. And I want to use this slide from Kendra Rumbau, which shows you signaling, chemical signaling, around the living world. And it's striking that there are bioactive small molecules that are involved in signaling between Monera, the bacteria, the fungi, the plants, and animals. And these small molecules, many of them look like antibiotics. The point is that they are all bioactive, and they are bioactive at low concentrations. And so here we have the adrenergic hormones. Here we have a variety of compounds that are active in quorum sensing and similar agents, similar processes. But in actual fact, antibiotics would come into this class. And antibiotics would come under this class because they also interact with animals and with fungi. So antibiotics are a much larger class of signaling molecules. I'm calling them antibiotics now because you understand what I'm talking about, but I don't have a better word. But there are a lot of molecules made by microorganisms that will interact at different places during this particular cycle. And there are many kinds of signaling reactions. This is not all of the signaling reactions that are known. For example, we now know that there are gaseous reactions that can transmit signals between bacteria. There's light, there's a nanotube, which is a chemical signal, and electrical. The two best known cases are where you have a signal emitter that produces a small molecule. This will either interact with a membrane or cytoplasmic receptor and activate a two-component system, as is the case with quorum sensing or the homoserine lactones, or in the case of the antibiotic-type molecules, the small molecule actually has a macromolecular receptor. The receptors for antibiotics are ribosomes, RNA polymerase, DNA polymerase, the cell wall polymerases. This is a different kind of signaling response. The main message is that the signaling response is a specific transcription response. There are many examples of this kind of thing. This comes from Diane Newman's work showing that the phenazines modulate colony morphology in bacteria. Colony morphology is a beautiful way of looking for the effects of low concentrations of compounds. Here's another example from Kenji Ueda, where they show, with Beppo, he showed that a particular strain or a product of a strain placed at one particular spot in a series of other bacteria, you find that you activate the production of an antibiotic here, or here, and here you're bringing about changes in morphology. So these small molecules are doing a lot of things. Morphology, they're producing antibiotics, biochemical changes, you name it. And we've been working recently on things which are doing something a little bit different. We were tempted by, talking with Mike Thread a few years ago, before he came out with this slide, showing that the interactions of environmental signals can be cross-interactions. So we can't always think of these things as these small molecules as doing one thing and not another. And they can be given different responses by the presence of other small molecules. And so the chemical signaling is also crosstalk, and we must not eliminate that. So this is the kind of model that Mike came up with, and we can see crosstalk. Now this is what I mean by looking at crosstalk. Here we're looking at plant hormones in bacteria, asking the question using Lux reporters in Salmonella or E. coli, and asking the question, do plant hormones activate bacterial responses? And the answer is very, very clearly yes. Here we have abscisic acid responding to a variety of different reporters. Also methyljasminate, both signaling or hormonal type molecules in plants. This is inter-kingdom signaling, exactly the kind of thing that I showed you earlier, but this is between bacteria and plants. And we've gone on to study this in other ways, and we, as you probably know, Mark Light and Primrose Freestone came out with this great idea a few years ago that there was microbial endocrinology. And microbial endocrinology involves interactions of bacteria with adrenergic hormones. And we first began to ask the question, what about the effects of antibiotics plus adrenergic hormones on bacteria? And we see that the adrenergic hormones, in this case, sorry, these are the plant hormones. The plant hormones definitely stimulate activities of the transcription response in bacteria. So this is an antibiotic-hormone interaction. Here's a case where we've been looking at the effects of plant hormones or bacterial hormones on morphology and swarming morphology in bacteria. So here we have a group of spots of bacillus strains on a plate, and this one has indole acetic acid in the middle. And I hope you can see that in this particular case, this particular bacillus strain has swarmed out a short distance. That same strain with indole acetic acid in the middle is swarmed out like this. So the effect of hormones on bacteria must not be discounted. And the effect of hormones on animals must not be discounted, but they're interacting. There is clearly a lot of competitive and interactive signaling within microbial communities, and within microbial communities and the human gut. There's absolutely no doubt of that in my mind, that we're looking at things which are a form of homeostasis between different organisms. Now, finally, I want to come to the question of if we can find new antibiotics, what can we do with them, and how can we get them? I've told you already that you can find bioactive compounds from nature very easily. You find them by using what are presumably their natural activities. You're not asking for antibiotic action, you're asking for activity as transcription modulators or as signals. So, a few years ago, Stu Schreiber came up with this notion of the revised central dogma, that one should not forget bioactive small molecules. I happen to have put penicillin up here, but I could have put any known antibiotic. And there are millions, billions of small molecules that we should be able to work with. And that's illustrated on the next slide. This is a modification of a slide by Nikos Kyperidis. He put that up a few years ago, and he wanted to give the enormity of the numbers of bacteria, the numbers of molecules, etc., etc. And if the bacteriophage is the most abundant, if the number of microbes on Earth is that number, then we would estimate that at least that many compounds would be made. And when I say that, I'm making it with the knowledge that there are bacteria that make bioactive small molecules that we did not suspect. For example, Staphylococcus aureus, a nasty bug. Nobody ever suspected that it produces bioactive small molecules. Staphylococcus does, and many other small strains do produce bioactive small molecules. What are their activities? I don't know, but they certainly do activate transcription. So we have number of humans, it's pitiful. Number of stars in the universe, much smaller. The US national debt is trivial. So we have things that show us that the numbers of small molecules in the biosphere are enormous and critical. And they're very, very important. Now, how do we get at these small molecules? You can't always just grow the strain and get the product. You have to now use modern genomic approaches. You have to go into genome mining. And this is just a very simple outline of what one can do in terms of trying to isolate a small molecule from an environment. You have to start with a genome or metagenome sequence. Once you've got the sequence, you can scan for potential biosynthetic clusters. The situation is such now that if you have, in many cases, you can predict a structure based on the genome sequence of the biosynthetic cluster. What you also might be able to do is to, if you know the structure of the molecule, you can carry out virtual docking on collections of molecules that have targets which have their X-ray crystallography, their three-dimensional structure established. So you can do virtual docking. You can then, if you find something that will dock onto a particular target, you can then go to look for the molecule. Now, there are many versions of this scheme. This is a simple one, and it's the most ambitious, because you don't have to purify anything until you get down to here. So I was lucky that we were able to do something like this with a group of people at UBC. We were screening for cathepsin K inhibitors produced by lichens. We found streptomycete that had a potent inhibitor. We partly purified it, but we couldn't get it very pure. So what was done was to soak it into a cathepsin crystal, and after soaking it into the cathepsin crystal, the 3D structure was determined, and we were able to identify the nature of the molecule. So this thing can be done. It's expensive, it's slow now, but it's the kind of thing that we will do more and more in the future. So, finally, I want to tell you how I don't get excited very easily. But what I'm really excited about now is microbiology. I think it's at the greatest stage in its entire history. And that is, it's coming from genomics. Now, the problem is, we don't want it to become just genomics. It must be microbiology. We must not forget the biology part. But I think that microbiology is going to be able to do so many more things now and in the future than everyone would have ever conceived of in the past. I mean, let's face it, who would have thought that there were so many microbes in soil? Who would have thought that if you go into soil and you go to a depth of 2 centimeters, 2 centimeter tranches, that you would have different microbes in each tranche? These are the kinds of things that are exciting and wonderful. And then we have this whole question of microbiomes. The skin microbiome, if you look at 20 sites on the skin microbiome, they are all different. That is something which is amazing, but on the other hand, you may find interesting molecules there. I told you that Staph aureus makes bioactive small molecules. It might, Staph epidermidis probably makes bioactive small molecules. So there's a lot of chemistry, there's a lot of bioactive molecules that remain to be discovered. And we want to discover them because they are acting as signals. They are part of community structure. And as soon as we know something about them, we can use them. So I'm not necessarily in favor of sequencing everything in the microbiome, but I think you should be well aware of the fact that microbiome biology is going to change the way we do microbiology, and it's going to change the way we do medicine. And then finally, this is my current lab group. We moved into a small lab, which is a bit small. And you notice that I have my old Welsh jersey on. I didn't win it, I bought it. And finally, I want to show you now my microbes. In case you're interested, when I started in chemistry in 1953, I actually was working with a microbe. I didn't even really know it was a microbe. Somebody said it was a fungus. Anyway, I was growing it and isolating compounds from it. Then I didn't do very much until later I started working on the cell wall with Milton Salton, which was a great thing. And I don't want to go through all the rest of it, but I've been fairly indiscreet in terms of the microbes that I've worked with, but they've all served me well, and I love them. And I hope you love microbes also. Thank you very much.